In previous part, we discussed about glycogen metabolism and various types of glycogen storage disease along with mnemonics. Now, in this part, we'll discuss about the particular types of glycogen storage diseases. First one is type 1 or bone jerk disease. Bone jerk, I have told G for jerk and G for glucose 6 phosphate is deficient in this condition, and there are two types of it, type 1A and 1B. 1A is enzyme deficient and 1B is there will be glucose transporter deficiency, and it presents at 3 to 4 months of life. Glucose 6 phosphatase is an enzyme present only in liver, right? And in this condition it is absent, it is deficient, so liver cannot contribute to the blood glucose level in the circulation, because glucose 6 phosphate formed cannot be converted to glucose and cannot be liberated to the circulation, due to which there will be fasting hypoglycemia. The fasting hypoglycemia causes decreased glucose supply to the brain, causing seizure, and to the body parts lethargy, and there will be lactic acidosis, which is another prominent characteristic finding in this condition. To understand the lactic acidosis, we have to understand the Cori's cycle. In Cori's cycle, when muscle utilizes glucose to produce the energy in the form of ATP, by glycolysis pyruvate is formed, and further anaerobic metabolism forms lactate or lactic acid, which is then transported to the liver to again generate the glucose for the further use, right? This is recycling process. So the lactate formed converted to the is converted to the pyruvate and is converted to the glucose. When there is deficiency of the glucose 6 phosphatase, which is a reversible enzyme for reversible pathway in the glycolysis, pyruvate cannot be converted to the glucose, right? So when pyruvate cannot be converted to the glucose, it backups, right? When it backups, it leads to accumulation of the lactic acid. So lactic acidosis occur, and again the glycogen is not broken down, so glycogen it accumulates, leading to hepatomegaly and growth retardation. Here in this picture, there is a child with bone jerk disease. The features are doll-like faces and protuberant abdomen. The protuberant abdomen is due to hepatomegaly. Now 1B is associated with the neutropenia, and the treatment is frequent oral glucose. Oral glucose is given frequently, but fructose and galactose, they are restricted. Other form of carbohydrates are restricted because gluconeogenesis is also impaired due to lack of glucose 6 phosphatase enzyme. When it is absent, gluconeogenesis is restricted, and when we give fructose and galactose, these are converted to the product of or metabolite of glycolysis, ultimately converted to the glucose 6 phosphate. Now it cannot be converted to glucose, so further there will be aggravation of the metabolite and further aggravate the symptoms, so they are not given. Now type 2 or POMS disease, POMS P in the first and fourth carbon, so 1,4-glucosidase enzyme is deficient, and when there is deficient 1,4-glucosidase enzyme, it is a lysosomal enzyme present in the muscle, right, which breaks down unbranched glycogen chain directly to the glucose, right? We have discussed here. This unbranched glycogen chain is acted upon by alpha 1,4-glucosidase present in the lysosome of the muscle, and directly from the glucose surpassing all these steps. When it is deficit, when it is absent in type 2 GST, there will be accumulation of the glycogen in the muscle, right? When there will be glycogen accumulation in muscle will be enlarged muscle. There will be muscle inactivity, right? Decreased activity, decreased muscular function, leading to hypotonia. When there is enlarged muscle, there will be cardiomegaly, obviously. Heart muscle also increases, and enlarged tongue, another skeletal muscle. Now you can remember is heart is affected, which is characteristic finding. You can remember is Pomfrey's disease, pump, right? Heart act as a pump. Heart act as a pump in our body, right? So heart is affected. There will be cardiomegaly. Here is a feature. Here is massive cardiomegaly in case of cardiothoracic ratio is greater than 0.5 in case of Pompe's disease. Here is a picture demonstrating the hypotonia in a baby with Pompe's disease, which is due to the excess glycogen accumulation leading to decreased activity, right? Decreased muscular function. But there will be no hypoglycemia. Why? Because again in this pathway, let's come in this pathway. Here glycogen is broken down by the two the glycogen is converted to glucose by the two pathways. First is this one, right? Through the glucose 6 phosphatase. Other is this one through the lysosomal alpha 1 for glucosidase, right? This is absent, right? This pathway is not working, but this is working, so there will be no hypoglycemia in the Pompe's disease, right? There will be liver enlargement, but not due to the storage dysfunction, due to the heart failure. Congestive liver enlargement will be there. Now type 3 disease, Core's disease, C for Core's, CD, debranching enzyme is absent. There will be milder hypoglycemia because the these are the glucose molecules, and this is a branching, right? Glycogen phosphorylase, which is the key enzyme in breakdown pathway. This is the activity of glycogen phosphorylase is normal, right? So glucose is maintained in the circulation by glycogenolysis. Only the branching is only the branch is not cleft, right? So there will be milder hypoglycemia. Liver and muscle both are affected since the enzyme is present in both, and there will be no lactic acidosis. Now in type 5 or McArdle's disease, muscle glycogen phosphorylase is deficient. It is a disease of adolescent and adulthood. Within the muscle cell, there will be accumulation of glycogen because glycogen phosphorylase, the key enzyme in glycogen breakdown pathway, is absent, right? So there will be glycogen accumulation within the muscle. Same as here, there will be glycogen accumulation. Similar features, there will be muscle injury first of all, right? So there will be muscle decreased activity of the muscle, cramp, fatigue, weakness, hypotonia. All these features are present, and there will be the injury of the muscle leads to liberation of the myoglobin, right? Myoglobin is a oxygen carrying pigment in the muscle. So myoglobin liberated in the circulation is ultimately excreted through the urine, leading to myoglobin urea. Urine turns dark, which is after the exercise mostly.